This is Pepe. Pepe is a male frog. Frogs are amphibians, meaning they live in the water for part of their life and on land for another part of their life. Even though Pepe is a male, Pepe does not possess the male genitalia. So, how does Pepe reproduce? Frogs reproduce through external fertilization, wherein the female releases her eggs from her body into a body of water. Then, the male releases sperm to fertilize the egg to produce zygote. To know more about the process of frog embryology, stay tuned and watch the video.
After the egg is fertilized, the zygote nucleus then undergoes a series of mitosis, with the resulting daughter nuclei becoming partitioned off by cytokinesis in separate and ever smaller cells. There are 4 cleavage stages in frog embryology. The first cleavage is a 2-cell stage and is formed vertically. The 2-cell stage forms after the first cleavage, which shortly occurs after the zygote nucleus forms. The second cleavage is a 4-cell stage, which is also vertical and right angled with the first one. The third cleavage is formed horizontally and consists of asymmetrical cells. The 4-cell stage forms after the second cleavage furrows again and runs through the poles but at right angles to the first furrow. The 4th and 5th cleavage form 16-celled zygote, leading towards 32-celled stage. At the end of cleavage, the solid ball of cells gives rise to blastula, which consists of a number of blastomeres. The characteristic features of the blastula stage are the presence of a well-defined cavity called the blastocele. This is the beginning of the primary body cavity. The next stage after cleavage is gastrulation. During the gastrula stage, the 2-layered embryo formed by migration and rearrangement of cells blastula. In this stage, differentiation of cells occur from monoblastic to diploblastic, which is composed of 3 germ layers. The cells in the blastula modify themselves spatially to shape the 3 layers of cells in a cycle known as gastrulation. The endoderm eventually becomes the sensory system and epidermis. The mesoderm eventually becomes the muscle cells and connective tissues in the body. The endoderm eventually becomes the columnar cells found in the stomach-related framework and numerous inward organs. During gastrulation, the blastula overlays in on itself to frame the 3 germ layers, which is the ectoderm, the mesoderm, and the endoderm. These offer ascent to the inner structure of the life form. The endoderm eventually becomes the sensory system and epidermis. The mesoderm eventually becomes the muscle cells and connective tissues in the body. The endoderm eventually becomes the columnar cells found in the stomach-related framework and numerous inward organs.
At a particular district beneath the equator, the blastoderm cells accept an extended jug-like shape. They push toward the inside of the blastula. As the cells move further inside, an invagination occurs. An extending of the invagination brings out a depression called the archenteron or gastrocele. The opening of the archenteron on the outside of the blastula is known as the blastopore. The blastopore, bit by bit, turns into a crescentic shape. At long last, it becomes round. The district dorsal to the blastoporal opening is known as the dorsal lip. The lower edge might be known as the ventral lip. The surface cells transition to a few imminent zones of the embryo start to meander inside the blastopore. These inwandering of cells are known as involution. We also have cell differentiation. Cell differentiation is the process by which the frog embryonic stem cells begin to create different types of cells, which would in turn form different tissues and organs essential to the embryo's development. It is important to remember that all cells have the same DNA, but in this process, the genes in the DNA are being regulated. Most of the time, proteins determine which areas of the DNA code is used, which would impact the cell structure and function. For example, muscle cells and blood cells have the same DNA but have different functions. Organogenesis, meanwhile, is the process by which the three germ layers of the embryo, ectoderm, endoderm, and mesoderm, develop into the internal organs of the frog embryo, occurring as many times until it reaches full development. During this stage, the embryo is called neurula. The ectoderm forms epithelial cells and tissues, even neuronal tissues. During neural systems formation, cells found at the edge of the ectoderm are signaled to become epidermis cells by special signaling molecules called growth factors. The cells in the center form the neural plate, which is a key developmental structure as it is the basis for the nervous system. The neural plate undergoes a series of cell movements where the neural folds fuse and forms the neural tube. The anterior end develops into the brain while the posterior end develops into the spinal cord. After the neural tube's formation, the mesoderm on both sides will form the frog's connective tissues. The mesoderm is reorganized into groups of cells called somites, which will develop the frog's ribs, lungs, and spinal cord. The endoderm then forms most of the internal organs including the digestive system, lungs, liver, and etc.
Now that you know the intricate and complex stage of frog embryology, please stop killing the little creatures that you see along the road. Thank you for watching.